And now it's time to introduce our guest speaker. Dr. Thomas Schubert earned his PhD degree from University of Regensburg, Germany in 2012. He has published 25 peer-reviewed manuscripts and 3 book chapters. In 2013, Dr. Schubert joined the company Tubind, located in Regensburg, as a microscale thermophoresis application specialist. He successfully established a semi-automated MST platform, which allows standardized biophysical analyses of aptamer target interactions. Thomas is now CEO of Tubind. Under his direction, Tubind is now one of the leading service providers in this field of molecular interaction analyses based on the MST technology. The Creoptix team is proud partner with Tubind, helping them detect and quantify biological interactions in real time for high quality kinetic data. I present to you the kinetics expert, Dr. Thomas Schubert.
Thank you so much, Julie, for the kind introduction. I will share my slides now with you. Julie, I think you might, okay, perfect. I think that's it. It's my real pleasure to be here in this talk about kinetics, and thanks a lot to Creoptix for the invitation. It's a great pleasure. The outline of this, there's a problem, here we go, is presented here. So first I will have some few slides on Tubind, after these slides, who we are, what we do, and you can better understand what we do with the GCI, which is point 2. And then there's a showcase for the waveRAPID screening, which I think underlines what the GCI is capable of. Tubind is a provider for biophysical analytical services located in Germany. And we are experts in biophysics, and our mission is basically to help people in their projects whenever biophysics are needed. Biophysics is a rather generic approach, so you can apply it for different types of molecules and different types of areas. And we do so here at Tubind. However, we also have certain focus on specific 1 or 2 areas, which I will bring up in the course of this discussion as well. So this is the summary of our capabilities. So we think that science is quite important to understand customer projects, to basically have good knowledge in the team is important. So 50% of our staff has PhD level, the remaining staff has master thesis. We have a fully automated screening lab with compound handling, liquid handling, biophysical screening and data processing. We have 8 core technologies and a track record of several hundred contract research projects, which underlines our experience. And important for us is that we call ourselves to be experts in biophysics, and this means that we should really take care about biophysical methods. And to do so, we work close with technology partners, and we are certified by Creoptix and Nanotemper for being their partners, which is very important for us. We want to take part in development of biophysical methods, and this is an important point, being, for example, beta testers. This is basically what Tubind is doing, and you can, as I mentioned before, you can see here, we work in different main areas, and the focus today, also because of the showcase, is drug discovery, but we also work in fragment-based drug discovery, protein biophysics, antibody development, aptamer research, and RNA-based drug discovery. Within drug discovery, we work on different processes. It starts at sample QC, then hit ID, hit validation, and hit optimization. And our idea of working on projects is that we want to explore and apply a customized, a tailor-made set of technologies towards a specific project. So we don't think that it's a good choice to basically orientate a project according to the tools that are available. So a customer that comes to us has an open discussion and gets the right tool for its project and a right combination of tools, actually. And this is, I think, the way we should go. An important piece in our portfolio is the Creoptix WAVESystem, which we apply for hit ID, validation, and optimization. I mean, as you know, this grating-coupled interferometry device, WAVE, is studying kinetic information of molecular interactions, for example, of small molecule drug target interactions or antibody-antigen interactions. Kinetics are important, there's no doubt. These are very important parameters when developing molecules which bind to a drug target or to an antigen. In the next slides, I would like to show you a little bit about GCI. I would like to introduce you briefly to the technology. And before doing that, I would like to highlight two major principles that are out there to study signals from surface sensors. The one is the classical SPR, surface plasmon resonance, shown on the left side. And it uses changes in refractive indexes once an analyte is bound to the immobilized ligand. And SPR is focusing on specific spots on the sensor chip, so it's a local signal readout. Hence, the readout intensity is rather small, so the small signal is rather small. Waveguide interferometry as a principle it rather uses a readout that is across the complete sensor. This should be done by coupling of a reference beam and a sample beam and really be able to analyze the complete chip, which would grant you the higher signal. Technically spoken, this is really challenging. So to develop such a technology and also to be able to produce that, this is the critical point. And that is also the reason why SPR is the more common principle. Creoptix has mastered that issue, and by a clever way of grating and basically aligning and coupling the sample and the reference beam into a waveguide below the sensor, it's now possible to analyze the signal from the complete chip. You have two beams that are coupled and they form an interference pattern. Once an analyte binds to the immobilized ligand, the refractive index change translates to a shift in this interference pattern, and this is the basis for the readout that is done, which leads to kinetic information, and the recorded information is of high resolution and sensitivity. Between or besides other interesting measurement modes like off-rate ranking and others, wave delta allows you to have two interesting measurement modes called multicycle kinetics. This is a more classical approach, and the new waveRAPID module. Multicycle kinetics, as we know also from other such as SPR, analyzes each concentration of analyte independently of the next concentration. Basically, you analyze different sets of concentration, summarize the data to get kinetics KDs, but also equilibrium KDs. The waveRAPID has a different idea. RAPID stands for repeated analyte pulses of increasing duration. And indicated by these bluish boxes, this means you have one concentration of analyte, but you give it in pulses that have different durations, so it's getting longer and longer, the injection. And by doing so, you can nicely record the dissociation rate, and by a mathematical maneuver, the association rate is calculated, and you can get kinetic informations out of that innovative new measurement mode. Even though the x-axis is not completely comparable, you might already see the point that in fewer time you get more concentration points and get faster data by using waveRAPID compared to the multi-cycle analysis. Hence, waveRAPID is a nice tool for screening approaches. You get kinetic information already at the screening level, and this is quite powerful, as we all know. We really like about GCI that it's a very versatile tool. So we can apply this tool in 5 of the 6 main areas that we're working in, and we can work with this tool in 3 of the 4 major process items that we have. So it's a really, really versatile tool and an important tool in our portfolio. To demonstrate the capability of that device and about assays that we perform at TwoMind, I wanted to show you a screen using P38, a known kinase, and 80 known kinase inhibitors. First of all, why P38? Yeah, because it's a perfect model system. There are many, many crystal structures available. The signal transduction pathway is more or less completely understood. It's very known about everything about where P38 is involved in cellular processes like proliferation, metabolism, and so on. And there are a lot of described small molecule inhibitors. So this tool is a quite nice model system, and that's why we have used it. And the idea about this screening is simply that kinase inhibitors very often have off-targets. So they are designed to bind a specific kinase, the on-target, but suddenly they show off-target effects. And we were interested in the fact if out of those 80 known kinase inhibitors, there are some which have P38 as off-target. Of course, we had a kind of expectation that this will happen, and I think I will not spoil it too much to say that this indeed happens. So there are some kinase inhibitors that have P38 as off-target. This is the screening process. How does it look like? So we start with a short sample QC of P38, and then the assay is developed. We start with a ligand immobilization establishment, followed by positive control establishment. And after we have done that, the ligand library has to be spotted into the different formats using lab-side echo spotting, either into the waveRAPID format or into the hit validation format with multicycle kinetics that we aim to do. Here's the sample QC step. So we have used nano-DSF, which is a thermal unfolding measurement type, which tells you the thermal stability of the protein of P38. And we have done that in without DMSO and with 5% DMSO, which was the assay concentration. And here on the left side, you see an unfolding profile in the ratio 350 to 330 nanometers. You can see that in the dashed lines that there's clearly 2 very nice unfolding transitions. And we end up at 51 or 52 per degrees Celsius, depending on the presence of DMSO. So this tells us the protein is quite fine. Just as a comment, there's also other options to do more QC. In this case, it was not needed. Coming to the ligand immobilization, which is one of the most important steps in the complete process. Here, one should really think about the target and what the target is about, what it may be harmed of, and what it tolerates, and so on. And there are several aspects that we need to consider for P38 immobilization. So first of all, it's known that P38 is sensitive to pH shifts. And this is a problem for classical amine coupling. So the concentration via a pH below the isoelectric point of a protein may simply harm the protein when it's on the surface or when it gets attached. So this is a problem that we need to overcome. And therefore, we have chosen a combination of his capturing the protein and then amine coupling. This allows us to do the immobilization at pH 8, which is for P38 obviously the better choice. And the second thing that we need to consider is the fact that a lot of kinase inhibitors are active site binders. P38, as quite a lot of kinases, contains an important lysine in the active site. And if we do amine coupling, we do that via lysins. So there is a chance that potentially we might harm the important lysine, which is in the active site. And to overcome that, we have saturated P38 with an active site binder before capturing and coupling, and also during. And in this way, we can protect the lysine, and after the immobilization is done, we can simply exclude the active site binder by injections of buffer, washing it out basically. So this is the immobilization protocol that we have used for this his capturing amine coupling protocol. So we activated the nickel surface with the nickel ions. Then with EDC NHS, we activate the surface for the amine coupling, and then injected P38 in repeated injections for his capturing amine coupling to a density that we calculated upfront. Passivation of the activated carboxymethylene groups is with ethanolamine. Nickel ions are removed by EDTA pulse because we know that nickel might have an effect on compound binding, and we want to get rid of it before the screening. So in the end, we come up with a nice coupling efficiency and density. So we're really happy about this. And later positive controls also showed us that the ligand activity on the surface is determined to be roughly 80%, which is quite nice. We are really happy about that. Just as a side note, we also tried the classical standard amine coupling without blocking of P38 with a compound, and we ended up with below 10% of ligand activity. And this basically already underlines that a thorough, thinking about ligand immobilization is really needed to get proper results, specifically for a screening approach. Next point is, of course, after ligand immobilization is the positive control establishment. So we've started with a compound called SB203580, which is described in a publication using SPR multicycle kinetics to have a kinetic KD of 12 nanomolar. And in the GCI, we end up with 37 nanomolar in the KD. Out of the kinetics equilibrium KD comes up with 45 nanomolar. So quite comparable, I would say. The WaveRAPID shows us a kinetic KD of 41 nanomolar, also quite in line with the multicycle kinetics. To judge the quality of data, it's also quite important, specifically for multicycle kinetic analysis, to check the off rates and how they follow the dose response. And as you see here in the middle graph, the dose response is nicely followed. So we can be quite happy about the quality of those data. It's quite homogeneous also how the curves come together. The second control that we tried was of the same series SB, but 2021-90. In the multicycle, we end up with 40 nanomolar kinetic KD. From WaveRAPID data on the right, we end up with 49 nanomolar, also comparable. The difference here is that the quality is not as good as for the control before. Hence, we decided for the control before. Nevertheless, just to give you also another control that we tried, it's BURB 796. And interestingly, this guy here has a very slow off rate. As you can see, it does not get rid of the surface, at least in that time frame. And it's also described to have a half-life of something like 23 hours. So this is not a proper control for what we want to do. It's a binder, but it's not a proper control. Okay, so to be sure that the screening that you have done is really valid and that the results can be trusted, We, of course, measured also the control during the screen, in total 9 times. And there are 2, let's say, factors that can tell you if the assay is properly performed or if the protein is active for the complete process of the screening, which is surface activity and the binding affinity of the positive controls. For both, we are quite happy. So surface activity dropped mildly from 80 to 50%. 50% is still quite high, so we are really happy about that. And even more important, the binding affinity of those 9 measurements, as shown here, was basically not changed. So the average affinity of all measured controls was 61.4 plus minus 2.3 nanomolar. So we can be happy and satisfied that the assay is valid and we can trust the data that we have produced. So out of these 80 compounds, we found 87% non-binders, 4% promiscuous binders, and 9% hits. And I will walk you through examples of each of those categories now. So first, let's start with non-binders. There are 3 examples, waveRAPID data in the top, raw data in the lower part. And in none of them you can see any dissociation. This means there is no binding. This, I think, explains enough. And then we have the category promiscuous binders. Here, these guys, they are clearly non-specific binders to the matrix. As you can see in the raw data, both bind reference and P38 surface in the same extent, meaning they simply bind the matrix. It's unspecific. And then we have a quite interesting one. So this one, we classified it as a non-specific binder to the protein. How do we come to that conclusion? I mean, in the first place, it binds to the P38 surface and not to the reference, or not too much. Meaning it could also speak for a normal binder. But if you look at the wave data, in general, there's a very slow on and basically no visible off. So this doesn't really fit to a classical binder. In addition, when you compare the signal of that experiment to the overall screening data, the signal is rather high. So therefore we conclude that it's potentially a non-specific binder, and we think that this compound might aggregate, but the aggregates are present in rather low concentrations and bind unspecifically to the protein. This is a hypothesis, sure, no proof, that's clear. We simply need more experiments, but I think this hypothesis could make sense. Then we have a bunch of different binders, as you can see here in waveRAPID data. In all of them, you can see a nice dissociation in the waveRAPID. And we have taken those guys and validated them with multicycle kinetics. So just as a very important information upfront, the screening approach with waveRAPID was done at 50 micromolar. So this is the concentration that we have to keep in mind. And in the multicycle kinetics, we basically adjusted the concentration according to the wave data. And you can see that there is other concentration used on the left side. So it's for the upper one, it starts at 200, for the lower one at 100. Nevertheless, the data look quite fine. So we end up with low micromolar KD kinetics and equilibrium for both of those guys. Again, when you zoom into the dissociation follows nicely the dose response. So nice data. Same goes for this colleague here, triple-digit nanomolar affinity, also nice data. And then there are some quite interesting guys coming up. First, this one here. We have classified it as binder, but it seems that this compound is aggregating from a certain concentration, meaning concentration-dependent aggregator. How do we come to that conclusion? In the left part, so the multicycle data, you see a black line, which is the expected Rmax, which is calculated by the molecular weight of the compound and the surface activity and density. And you can see that quite a number of data signals are above this threshold. And this already indicates that it's definitely not a one-to-one situation, and it's not stoichiometric. Furthermore, also the shape of the curves and some other aspects quite indicate that this is an aggregation problem here. Still, we think that this is a binder because when you look at lower concentration of this compound in this assay, it quite follows a one-to-one interaction and it's still reversible. So, of course, there's no saturation reached, and therefore we call it an apparent kinetic KD of roughly 70 micromolar. But I think that's an interesting candidate, and it tells us what kind of information you can get out of GCI. But I think that one is maybe even more interesting. So again, I want to remind you that we have screened at 50 micromolar, and waveRAPID has identified this as a hit, which is for that concentration also, it's true. But if you have a closer look, you can see that there's again a problem with aggregation, which you can see in the multicycle kinetic data on the left with the highest concentration. At least there's some weird things going on, but very obviously it's in the raw data. So here the signal increases quite intensely with the concentration. And in addition to that, it's clearly obvious that the molecule at high concentration binds to the reference surface and the P38 surface. Same goes for when you look at smaller, low concentrations. It's kind of reversible, yes, but it's still multiphasic and very likely non-specific. And this means we have classified this one as a non-specific binder and also concentration-dependent aggregator. Again, we have a kind of a theory without any proof. We need to do a lot more experiments, but if you look at the structure of this molecule, it looks like a lipid, like a Tenzit, like, detergent. And it could be that this molecule forms kind of micelles at a certain CMC or above a certain concentration. And those micelles could then potentially aggregate and bind to the surface of which is presented, whatever it is, so reference or P38 surface. Again, theory, we need more experiments, but I think it's interesting that what kind of information you can get out of such data. So to sum up, I think it's important to say that we have a nicely established assay. We found interesting hits. We found off-target, P38 to be off-targets of some kinase inhibitors. We found non-binders, promiscuous binders. But as well, it's important about to say what's the consumption we had, how many chips do we need, how long did it take us, and so on. And here's a little summary for you guys out there. So in the sample QC, we used 5 micrograms of the protein and took us roughly a day. In AcidF, it's 10 micrograms of protein and a microliter of the control compound stock. We consumed a chip in 2 days. Same goes for hit ID. Also, again, 10 micrograms protein, 1 microliter of the control compound stock, and 2 microliters of the inhibitor stock. Again, 1 chip, 2 days. Hit validation, another 10 microgram protein, 1 microliter again of the control compounds for validation, and 6 microliters for the inhibitor stocks. Also 1 chip, 2 days. And to sum up, for that screen, it took us 35 micrograms protein, 3 microliters of the controls, 8 microliters of the compound, and 3 chips, 7 days. The screen contained QC, assay development, hit ID, and also hit validation. We are big fans of the GCI at the Wave because, as I mentioned, it's a very versatile tool. So first of all, it has different measurement modes, the comprehensive multicycle kinetics, HDS, off-rate ranking, so on. For large molecules, it can also be applied in complex lysate, serum, whatever bioliquid. Big pro is that it can measure even fast kinetics and tightest binders. This is quite impressive, what we have learned in the past. It can be applied in quite a broad range, from small molecules to large particles. And one thing that is really important is that it has different immobilization strategies, and as SPR protocols can be directly transferred to GCI, which is really cool. Bringing us to the more or less last slide. If you want to learn more about us, about the technology or technologies, please contact us, come to our homepage, or directly write to us. You're always welcome. We are happy to answer any questions. Bringing me now to the Q&A session. And from here, I would like to hand over to Julie again. Thank you a lot for your attention. It was my pleasure.